Hi, my name is Elle Picard, and I'm a ninth grader at Washington Liberty High School, and I want to thank you for taking the time to listen to my presentation today. In 2015, my family and I decided to sponsor a young boy from Africa. Where he lives in Bamwalukani, Uganda, it's difficult to get safe water for drinking, cleaning, and cooking. For many places in the world, this is the reality. Bacteria such as Salmonella, Escherichia coli, Shigella, and Campylobacter cause many diseases and illnesses such as diarrhea, cholera, dysentery, typhoid, and polio. According to statistics from the World Health Organization, at least 2 billion people drink from a contaminated water source, and about 500,000 annual deaths are caused by these waterborne diseases. I believe that clean water is a basic human necessity, so I conducted this experiment because I wanted to identify effective and inexpensive methods to filter bacteria from water. Bacteria are single-celled microbes, and there are over 30,000 species. Different types of bacteria do many different things. Some bacteria are good for you and beneficial to your health. However, most bacteria are bad for you and can make you very, very sick. Bacteria reproduce by binary fission, a process in which the bacterium divides into two identical daughter cells. And when the conditions are favorable, such as right temperature and nutrients, some bacteria like Escherichia coli can divide every 4 to 20 minutes. This means that after a mere 8 hours, this number would be the number of bacteria would be at 16 million. And because of bacteria's rapid spread and prevalence, it's very difficult to get rid of. But high temperatures, high alkaline or acid levels, and strong light exposure, including UV light rays, can kill bacteria. I researched several different types of decontamination, including filtration through a coffee filter, boiling, chlorination via bleach, and solar sterilization. And the most useful sources for finding information on these home methods were government websites that provided information on making water safe in an emergency, and many organizations that helped developing countries gain access to clean water. So I found that coffee filters could be used to filter sediment and debris from water in order to make it more potable. Chlorine, when added to water, forms a weak acid called hypochlorous acid. And since the cell walls of bacteria have a net negative charge, the hypochlorous acid with a net neutral charge is not repelled and can invade the pathogens. And once the acid is inside the bacteria, it damages the structures of proteins and causes the microbe to die. I also found that in many studies, UV light was a very effective source for purifying water. Because UV light passes through the cell wall of the bacterium and permanently alters the microbe's DNA, making it non-viable. According to the Centers for Disease Control and Prevention, boiling is the surest method to kill disease-causing organisms, including bacteria. Boiling water uses heat to damage the structures and processes inside the bacteria, and the pathogens start to die. My hypothesis was that boiling water would kill the most bacteria, and my null hypothesis was that there would be no change between the groups. Each level of independent variable had 6 trials, and the dependent variable was bacteria per milliliter. Some of the constants were the amount of water in each container, incubation periods for the test strips, and the disinfection of all materials prior to testing. Some constraints I came across when designing the experiment was that the testing kit I had was pretty limiting. For example, the key was a linear scale of 1 to 5 different colored growths that the strips could be, and each color had a corresponding range of bacterial numbers. Prior to experimentation, all containers and bags for testing strips were labeled with the given group in trial. Then all materials and equipment were sterilized using a spray bottle filled with 91% isopropyl alcohol. And with 1 large bucket, I collected 3 liters of stream water from a tributary that drains directly into 4 Mile Run. 500 milliliters of water were put in a flat baking dish and covered, and then left in the sun for 6 hours. Then 500 milliliters were cooked on medium-high until they hit a rolling boil, then left on that rolling boil for 2 minutes. Then 500 milliliters of water were strained through a coffee filter that was secured around the top of the container. And using a ratio of 1 drop per 500 milliliters, household bleach with 6% chlorine was added into the water via pipette, and this amount is still safe to drink. Then I used 30 Waterworks bacteria growth check test strips to dip into the 5 containers of water, so 6 strips per group. And the strips were sealed into individual Ziploc bags and left to incubate for 48 hours. I then took a reading from each one of the 6 trials and averaged this data. The graph is the mean results for each group, and the indicated orange breaks scale the graph accordingly. As you can see, the control group had 340,000 bacteria per milliliter. The coffee filter and sunlight groups had a little over 167 million bacteria per milliliter. The boil group had 83 bacteria per milliliter, and the bleach group had 50 bacteria per milliliter. The chart below indicates averages, standard deviations, and individual t-test results compared to the control for each group. This top chart is the ANOVA results for the experiment as a whole, and again are just the individual t-test p-values for each level of independent variable. From our ANOVA test, we got an overall p-value of 0.0000, which means that the data can be determined to be statistically significant, and these findings can reject the overall null hypothesis, as the boiled and bleach groups had notably less concentrations of bacteria. The p-values of the boiled and bleach groups were both very significant, as they were well under 0.05. On the other hand, the sunlight and coffee filter groups were not significant, with both of them having more bacteria than the control. For the coffee filter, there could have been extra bacteria within the filter itself that further contaminated the water, which would explain why it's higher. And since sunlight is an uncontrollable circumstance in any it's an uncontrollable variable in any circumstance. It's not always reliable. So UV light also takes time to purify, with a minimum of 6 hours to be considered safe. But if these solar rays aren't properly reaching and killing the bacteria during this time period, the bacteria just has more time to reproduce and proliferate. So this could be why the sunlight group has a higher concentration than the control. Something unexpected was also that the two groups, sunlight and coffee filter groups, both ended up having the same number of bacteria, although, as I mentioned before, that was due to probably impreciseness of the testing kit. But overall, the bleach group was the most effective method with the lowest amount of bacteria, and the boil group was the most consistent with the lowest standard deviation. The strengths of my data were the differences that these two groups had compared to the control, the UV light group and the boil group. And the limitations of my data were, again, the impreciseness of the key, which influenced the exact numbers of the data. And to limit these constraints in future experiments, I'm going to use plate culturing to count the bacteria and get very accurate data. With plate culturing, you can also look at specific types of bacteria. So that would be helpful to see if there was any correlation between method of filtration and type of bacteria. I would also study using UV light to kill bacteria, because although my experiment was unsuccessful in finding a way to do that, most of the studies I read reflected the idea that UV light could be very useful. And the most important thing I learned from this experiment was that either boiling water or adding a drop of bleach into it would work to effectively remove bacteria in emergencies. And I'm going to take these findings and experiment with them in upcoming years, and I will continue to find the best way for people to get clean, safe water. These are some of the sources I used in my experiment, and that's it. Thank you.